So it says, you are holding a small plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Usually you would create this by, and we'll talk about this later in class, using enzymes called restriction enzymes. But you could also use things like CRISPR, so anything that can cut DNA into pieces. Then the problem is you'll have a mixture of pieces of DNA, and you can't see any of this. It looks like clear liquid, but it's got DNA in it. How do we actually figure out what's in there? And that's what this technique does. So your job is to figure out what those links are. How will you do it? So here's our pieces of DNA. We're going to line them up by lengths. If DNA strands were as big as your shoelaces, you could sort them by hand into groups and measure them. But of course, they're not, and they're tiny, and you can't see them. So here's our tube. This is called an Eppendorf tube. These little numbers right here, these are actually measurements. So this is in milliliters. So this is 1.5 milliliters, 1 milliliter, a half a milliliter, so and so on. So usually most of these tubes will hold about 1.75 milliliters. These are basically the workhorse if you wanted to work, for example, in a biochemical type of laboratory or any kind of DNA lab. This is the kind of stuff that you use. DNA strands are molecules that are so tiny you can't see them even under most microscopes. Is there a way to sort and measure DNA strands in your tube even though you can't see or touch them? There is. It's called gel electrophoresis, pronounced electrophoresis. Press forward to continue. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How does this work? It's a little more complicated when you have to do this with proteins. But basically this right here, this is a micropipette, or a pipetteman. That's actually a name brand. This is the microtube right here. This is the kind of setup labs all across the country, all across the world, that are working with DNA, that are working with proteins, that are doing any kind of biotechnology that we've been discussing. This is the kind of thing that you would see. This is what people do. This is how biotechnology is done. So this is a gel. So a gel, a gel is a filter that stores DNA strands. It's a sponge. It's like a sponge made of Jell-O with many small holes in it. So it's got little holes, and basically the longer the DNA fragment, the harder it is to get through this matrix. So if they're really long, they don't move very fast. If they're really short, they move faster. So this is how you can actually separate the pieces of DNA. So we add the samples. We place DNA samples into holes at one end of the gel. We call those wells. And we'll show you how we actually make this. So electrophoresis is how we push DNA strands through the gel filter. We add an electrical current that makes DNA move. DNA is negatively charged, so it's going to be repelled by this negative charge and attracted by the positive charge. So it's going to move from here to here. So from the well, the DNA, like I said, is going to move through here. The smaller they are, the faster they move. Short strands move through the holes in the gel much more quickly than long strands. Over time, the shorter strands in the sample will move farther away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, DNA strands in the sample sort themselves. So this is basically how you can separate these, because at the end, you cannot see an individual strand. So what you will see will actually be groups of strands of the same length. Staining the sorted groups of DNA makes them visible to the naked eye. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. So these little orange things right here, we call these bands. So a band is basically, this is a population. It's a group of DNA that all have the same length. These colors we'll talk about, these are due to stains. Now this video, this virtual lab is a little bit old, so they're going to use a stain called ethidium bromide. There are lots of newer stains that aren't as, they don't require UV light. They actually have different colors. But this is actually what the colors would be. They're kind of an orange color. If we have enough time in the lab, I may actually run one of these gels for you so you can see it. I always thought it was really cool to see the bands actually glowing because they glow under UV light. It's pretty neat. Okay, so what are we going to do? We're actually going to, well, okay, we're not actually going to do this, but we're going to do a virtual lab. So step one, we're going to make the gel. We're going to set up the gel apparatus. We're going to load the DNA sample into the gel. We're going to hook up the electrical current. We're going to run the gel. Then we're going to stain it, and we're going to analyze the results. So now it's your turn to run a gel. The scientist slang for gel electrophoresis. Run the gel, run the gel. Yeah, we did lots of that. Have done lots of that. So follow along with the steps shown above. Okay, so step one, we got to make the gel. So we go into the laboratory. My laboratory didn't really look like this. So here's the equipment we need. Here's what you need to make a gel. Powdered agarose, that's the stuff that will actually melt and make into the gel. Buffer, which is a substance that basically maintains a proper pH, if you remember way back when we talked about that. You need a flask to put this in, a microwave, the gel mold, and the gel cone. Still got a little cough. Making electrophoresis gel is a lot like making Jell-O, but you don't want to eat this type of gel. And on my microwave that I use for this, there's a big sign that says no food. Okay, so here we've got our agarose. Now the one thing that they're skipping here is you have to weigh out the amount of agarose. Normally you're going to control what the percentage of the gel is, depending on the fragment size. The larger the fragments you have, the lower the percentage of gel you have. Usually it's about 0.7% to maybe somewhere about 2 or 3%. If you're working with really small fragments, you could use, a 3% gel. And your gel will be thicker and heavier depending on how much agarose you actually put in. So we click on this. Click over here. Ta-da! Now we have to add the buffer. So we're going to add our liquid buffer. Now this is saltwater buffer. Usually you can use different types of buffers. One of the buffers is TAE, which is tris acetic acid, EDTA. There's also TBE, but those are the two major types of buffer. Just called it saltwater buffer. And again, you have to measure that as well to get your percentage correct. We've loosely placed plastic wrap over the top of the flask to prevent the liquid from boiling over. Open the microwave by pressing the open button. Place the flask containing the mixture and the buffer and agarose mixture inside the microwave. Heat the mixture until the agarose melts into the buffer. So you open the microwave, and this is how I did it. This might take a minute or two. Now, the one thing they skip that I usually do is I will swirl this and make sure that all the little chunks of agarose are melted. So making sure that it's smooth. You don't want to swirl it too much because you can get a lot of bubbles inside of it. You don't want that. But anyway, one thing I can tell you, there was a story. I swirled one of these flasks once, and I had microwaved it a little bit too long. And if you microwave it too much, sometimes it can be really close to boiling, but not actually boiling. So I swirled it, and what happens, it suddenly started boiling, and it shot out of the top of the flask, came up over my head, landed on my head. That actually happened to me in the laboratory. Now, fortunately, by the time it had gone up in the air and landed on me, it wasn't really hot anymore. But that's why you have to be really careful. Anymore now, when I swirl it, I'm very, very careful. So we're going to pour it into our mold here. Now, usually you also want to let it cool down a little bit before you do this. And when I do it, I always have the comb in ahead of time. But you can also add the comb in later. So we pour it in. We let the gel cool and solidify. This usually takes half an hour, but we'll speed up the clock for you. As the gel cools, tiny holes will form. They're left by the teeth of the comb. And you can kind of modify your comb. Sometimes some combs are big and wide, some are narrow. You can control how many wells you're going to have in a gel and how large they are by your comb. Carefully remove the comb, leaving empty wells for DNA samples. Your gel is now ready to run. There we go. Now, to actually run the electrophoresis, you cannot use pure water because pure water actually will not conduct electricity. So instead we use the buffer. Usually this is exactly the same buffer that we use to make the gel. So it says, now it's time to set up the electrophoresis box. You will need the gel you just made, an electrophoresis box, and another bottle of buffer. This one's kind of handmade. Any more, you can actually buy them. So we're going to press forward, read the instructions. Pour the buffer into the electrophoresis box. So we're going to pour that in there. Place the gel, still in the mold, into the electrophoresis box. We have removed the tape from the ends of the gel mold. The gel should be barely submerged in the buffer. Again, the modern structures usually don't actually have to use tape. But if you were to actually just build one of these yourself, you can use tape. And some people actually do this in their garage, so it's a thing you can do. The buffer conducts electrical current from one end of the gel to the other. It will also keep the gel from drying out during the experiment. Because if you run this with a really high voltage and high current, you can actually melt your gel if you don't have enough buffer. So we're going to pick this up. I'm going to put it in there. Now we have our samples, and we've got our micropipetter. You're ready to load the DNA sample into the gel. Here's what you need: loading buffer, the tube of DNA, the DNA size standard, micropipetter, electrophoresis box containing buffer in the gel, and pipette tips. So pipette tips over here. Now I do things slightly differently. They show the DNA sample. You just add the loading buffer to the DNA sample. I actually get a piece of parafilm, which is basically wax, and I put my loading buffer out there, and then I mix my samples while they're on the parafilm. But this will be a little easier as a video. So we put a tip on our micropipetter. With a clean pipette tip, use the micropipetter to suck up some loading buffer, then add it to the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if you tried to load it directly into a well. The loading buffer contains a dye that makes the sample easy to see. It's also slightly goopy, meaning it's got some, I forgot what it's called, but basically it's got a heavy substance in there that weighs it down. This makes the DNA sample thicker so that it will drop into the well instead of floating away. The DNA size standard already contains loading buffer. So we're going to do that. Now we're going to add it to our DNA sample. Next, you will use the micropipetter to transfer the DNA sample from the tube to the well of the gel. First, suck up some of the DNA sample into the pipette tip. We're going to suck that up. Now we're going to add it to the gel. Eject the DNA sample from the pipette into the first well of the gel. Now, to actually do this in real life can be a little tricky because you can sometimes miss the gel. You can also puncture the gel if you push too far down. So it's not necessarily as easy as this little animation makes it, but ta-da! Nice job. In real life, loading the sample into wells takes some practice, so don't be disappointed if you miss your target the first few times. See, they know what I'm talking about. Now we're going to load the size standard. Now usually when I do this, I load the size standard first. Using a clean pipette tip, use the micropipetter to suck up some DNA size standard. Okay? Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths. Running it on the gel will give you a reference by which to estimate the lengths of your DNA strands in your sample. So we also sometimes call this a ladder. And you can buy these commercially. And they'll actually give you a map of what they're supposed to be. It's time to turn on the electricity and run your gel. When you turn on the power, the black end will generate a negative charge. The red end will generate a positive charge. Together, they will pass current through the gel. DNA has a negative charge. I mentioned that already. To move DNA through the gel, you must put the black cord, negative charge, closest to the wells. So press forward. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Click the location on the power supply where you want to plug the black cord. Now, you don't want to do that backwards. You might think this is easy. We'll just plug in black to black. The wells have to be on the right side. So the wells need to be over here because basically the negative charge is going to push the DNA away because DNA is negatively charged. That's right. Now click the location on the power supply where you want to plug in the red cord. Turn on the power supply. So here we've got the voltage. This is a wimpy power supply. Usually I run at about 100 volts. Your gel is often running. Let's look inside the gel box and see what's happening. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are your proof that a current is running. So if this were real life, you would notice that one of these wires would have twice as many bubbles as the other. And that's because what these bubbles actually are is you're electrolyzing, meaning you're splitting water using electricity. So you're splitting water into hydrogen gas, and oxygen gas. That's actually the basis for things like fuel cells. It's one of the ways you can actually take electricity and essentially store it in different ways, is actually capturing those gases. But in this particular situation, it happens to be a side effect of doing electrophoresis. Okay, so we've got our wells. Repelled by the negative charge, the DNA moves through the gel towards the positive charge at the other end. Short DNA strands move through the holes in the gel more quickly than the long strands. Over time, the shorter DNA strands will migrate further from the starting point than the longer strands. We can't actually see the migrating DNA in gray, but we can see the blue dye from the loading buffer as it migrates. You've finished running your gel. Usually that takes about an hour. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your sample and estimate the lengths of the DNA strands. So, of course, I cannot see the DNA here. Right here you can see the dye. So usually when I do this, there'll be two dyes, so there'll be one here and one here, so I can kind of estimate about where the DNA is. But you can't see it until you stain it. First, you will need to stain the DNA in your gel using DNA staining solution. The stain is a chemical called ethidium bromide, which binds to DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see large groups of stained DNA strands. These groups will show up as bands in the gel. So what happens is this is ethidium bromide, so it actually gets in between the bases of DNA. So this is why it's considered a mutagen, meaning it can cause mutations. So when you do this part, you have to use gloves. Now, there are newer stains that are less toxic than these. There's like cyber green, there's gel green and gel red. There's a lot of them. They tend to be more expensive than ethidium bromide. I've used both. During my during my graduate school. Well, actually, I did some in undergraduate too. But we mostly use ethidium bromide, and it kind of looks the coolest. Okay. Drag the gel out of the mold and put it into the DNA staining solution. So here we go. Boink. So DNA can, it's going to stain. It has to diffuse into the gel. Takes about a half an hour to stain DNA in the gel. Sometimes it takes a little bit longer. We'll speed up the clock for you. Yay! Remove the gel from the staining solution and place it on the UV ultraviolet light box. So the UV is required because it excites the bonds that are stretched inside of the DNA that's in the gel, and that's what makes the bands glow. So they'll glow kind of an orange with the ethidium bromide stain. Turn on the UV box. Ultraviolet light from the box can damage your eyes. If you do this in real life, be sure to wear protective goggles. My UV box that I have in my lab actually has a little cover. So it's got a UV protective cover, so you can actually look at it without goggles. So it's kind of nice. So this is what we would see, is a bunch of bands like this. So this is our size standard. So here's the lengths in our size standard. So 6,000, 5,000, 4,000, 3,000, 2,000, 1,000. So notice the longer ones are closer to the wells where we started. The shorter ones are farther along. Now you can determine the approximate lengths of DNA in your sample. Compare the bands from the DNA sample with the bands of known length from the DNA size standard. Enter the estimated length in base pairs, so base pairs of the letters, A's, T's, G's, and C's, for each band in your DNA sample. Electrophoresis cannot tell exact DNA lengths of strands, just a good estimate. So give your best guess based on the DNA size standards. Press forward when you think you are finished. You check your answers. Okay, so this one right here is lined up with the 6,000. So we'd say, okay, this is probably 6,000. Now this one right here, this is between 4,000 and 3,000. So it's not going to be longer than 4,000, so we might say 3,500. That might be a good guess. So down here, we've got 1,000 and 2,000. And it's about halfway, but maybe we can say, so it's going to be bigger than 1,000, so let's say 1,400. See, all of your estimates are correct. They said this was 1,500, but might be a little, it looks like it's a little closer to 1,000, but all of your estimations are correct. The estimated length in base pairs of each band in your DNA sample is based on how they line up with the bands of no length in the DNA size standard. The top band is approximately 6,000 base pairs, the second is approximately 3,500 base pairs, and the third is approximately 1,500 base pairs. Congratulations, you have just run your own DNA gel electrophoresis experiment. Press forward to start over. So that is our experiment. So I want you guys to go through this, to watch it, to do it yourself if you can get the flash to work. And I will give you a quiz. I'll give you a gel kind of like this, and I'll ask you to estimate some sizes, and I'll ask you about the process a little bit. So anyway, thank you for watching, and I'm going to stop there for today.